Have you ever wondered what happens inside your body after you receive a COVID-19 vaccine? Or does the vaccination change the way your genes respond when you become infected with COVID-19 virus? So in this project, we are going to use RNA sequencing and bioinformatics tools to actually explore these questions by comparing the gene expression profiles from vaccinated and unvaccinated individuals who were affected by SARS-CoV-2 variant. So let's get started. Okay, so we are going to find our data from GEO. So what we are going to do is click our favorite browser, and then we are going to the NCBI, and that is the National Center for Biotechnology Information. So we are going to click here and then GEO datasets. Okay, so after doing some little bit of research, I found a perfect dataset that we can use. So we are going to use this specific dataset, GES 201530. Okay, so we hit on enter, and then the first search results that are going to come is our dataset. So this can be our perfect dataset because it contains 55 symbols which can work for our case. So let's see more information about the dataset. Okay, so I'm going to put a link in the description if you ever want to read this dataset for your convenience. Okay, so we are going to do dataset acquisition. That is simply downloading the information from the GEO. So basically they involve 3 steps. That is acquiring the dataset, downloading it, and preparing it. Okay, so this dataset is ideal because it was acquired in the same environment. That means the same test environment and the same platforms. The tools that we used were actually the same. So in this case, we used Illumina Novasec 6000. Okay, so in this bottom part, we can see we have supplementary files. These supplementary files, we have soft formatted family files and minimal formatted family files and series matrix file. Basically, the series matrix file contains information about the individuals, their age, their sex, and various other conditions like were they initially affected, do they have any side effects, various information containing the individuals. So if we go to this part and we click, we're going to have the soft file, the minimum file, and in this case we're going to ignore them. So we're just going to download the series matrix file and the row counts matrix file. Okay, so let's save it. Okay, then the next file is the series RNA-seq row counts matrix file. So the next step is differential expression analysis. This is typical bioinformatics pipeline, which is going to allow us to understand which genes were expressed and which genes were not expressed. We're going to find out how the vaccine has altered the level of gene expression in the human body. Okay, so basically in this, we are just going to statistically identify the significant expression or changes between the study group of the vaccinated people and the unvaccinated people. Now that we have the 2 files, that is the series matrix file and the count matrix file, we are first going to extract all the information from the files. Imagine these files as a piece of information that is no meaningful interpretation. Like, let's take for example, it's a hospital that has received a batch of blood samples. You cannot tell which blood came from which individual unless you're given a document that tells you this ID belongs to this person. So same as the count matrix file and the series matrix file. Unless we extract the metadata from these files, we are never going to know which genes belong to which person. So just for confirmation, if I open this file, we can see that it contains a lot of rich information, but we cannot generate any meaningful conclusion from it. And also, if I try to open the raw matrix file, we can see it contains rows and rows and columns of information. We don't have any relevance unless we do some statistical calculations. So what I'm going to do, I've written two scripts. The first script is called analysis. It is written in R program. I'm not going to be too much technical on how I've written the script. If you want to download the script and see it for yourself, it's in my GitHub link in the description. So let's just visually see. So since I was working in the background, I have already opened R Studio. So I'm just going to show you how the file looks like. So the first step is to just go to the working directory, and I'm going to open the R file, and I'm just going to show you. Okay, so let's open it. Just for context, I'm going to explain to you what is really happening behind the scenes. So basically, we start with data input, and then metadata pacing, and then data preparation, and then DSeq analysis, and then results. So this diagram shows you algorithmic explanation of the code that I have written. So all these steps are happening in a single click. As well, all the information about this is also in my GitHub link. Okay, I'm just going to start running the script and generate the results, because what we need at the end is a biological interpretation of the results that we are anticipating. So let's begin. Okay, so it's done. You can see it has completed. So later I'm going to explain to you all the results that you've get. But in the meantime, let's continue with the pipeline. Okay, so to continue with the process, we are now going to do the immediate next step, which is functional enrichment analysis. So before, what we have done is gene expression analysis. It doesn't tell us anything about the function and which pathways have been affected. It just showed us which genes has been expressed. Think of it as a reaction to a stimuli, right? What really happens is when the vaccine enters the human blood, right, there are some changes in the gene expression levels that is going to happen. Simply, it means there are some genes which are going to be in conjunction with the vaccine. But now we want to see, among the genes which have been upregulated or which has been expressed, does the vaccine really affect all the virus pathways? And as well, want to see what is the difference between a normal person who is not vaccinated and a vaccinated person. Okay, so without wasting time, I'm going to run the analysis. So let's continue. So like before, I also have written another R program, and I've labeled it as GEO. So it also contains lines and lines of code, and in a similar way, it is also going to run under the hood. So the process is done, and now we are heading on to results interpretation. Okay, so now it's time for results analysis. And you can see that comparing with the way we started, we now have a lot of files that have been generated. That is a typical bioinformatics setup. You start with 2 files, you end up with thousands of files. So if I go into this results folder, you can see that we have a lot of files as well that have been generated. So these are the plots, the visuals. You can see there are a lot of charts that have been generated from the analysis. So I'm just going to pick out the most significant analysis data that we can work out with this data that we have. So the very first file that I am very much interested in talking about is called the principal component analysis plot. earlier I've told you that we have more than 23,000 genes that were contained in all the 55 samples. The analysis shows us that in the healthy control, there is very minimal variation. Suppose we have a statistical average where all the 23,000 genes are lying. So the PCA plot is just picks out some component genes. Now you see that there are more outlying a number of genes from those people who have not been vaccinated. So we have one important interpretation as of now, that the health control is showing a consistent gene expression profile. Compared to those people who have not been vaccinated, it is showing that the data is much dispersed. There are more outliers. It's almost, if you combine PC1 and PC2, it's almost 55% of all the genes in non-vaccinated people are outside of the average value. So the next best analysis figure that we are going to do is the volcano plot. So the volcano plot is going to quickly show us which genes have a more significant expression than the other. So you can see from this graph, of all the genes that have been mapped, that is around 23,138 genes, only 445 genes have been upregulated and 74 genes have been downregulated. Among all the patients, if we compare the whole sample of 55 people, only 445 genes were upregulated and the rest were either downregulated or even unregulated, which means there was no significant change. So you can see that among these upregulated genes, we have genes like the IF127 or the IF114L. These genes they are showing a higher adjusted p-value than the others. That's another significant notice that we have made, and this is going to be explained further as we are analyzing all the results that we are having here. Okay, so the next result that we are going to focus after volcano plot is called the heat map. So basically the heat map is going to display the relative expression levels of each gene in every sample. So it is going to allow us to identify any patterns, any clusters, any similarities among both the genes and the samples. So by just looking at this, we can see that there are more number of blues than reds, and some clusters you can see from the left side. We can see this is where we get more number of reds than blues. I think this correlates with the volcano plot, because the volcano plot showed us that among the 23,000 genes, only 545 genes were upregulated, which means they were the significant genes which are being controlled by the vaccine. So the heat map is going in conjunction with the volcano plot. They're just showing us the biological variability of the genes and confirming that there are more genes in the healthy control which have no significant change. The very next thing, or rather let me say the last analysis that you are going to make in the differential expression analysis, because right now, remember, we don't have any significant information about which genes are responsible for the vaccine, what are they doing in the body, which pathways have been affected by the vaccine. We don't have that information. Okay, so now looking at the MA plot. So MA just means minus average. It is going to represent a log-fold change, showing us how much gene expression differs between the two groups. So the MA plot is a visual confirmation that more number of genes that are towards the zero, which are non-significant. And you can see from this one, without even telling you, that the upper part showing the positive change is showing the upregulated genes, because a log-fold change of positive means upregulation, and then a negative one means downregulation. So it's another confirmation. Okay. So as we are now moving on to functional enrichment analysis, we are now going to only focus on the upregulated and the downregulated genes. So among the 23,000, we are just going to focus on the 445 and the 474 genes which were upregulated and downregulated respectively. So let's move on to functional enrichment analysis results. Okay, so this result is is going to identify all the biological processes that were significantly represented by all the differentially expressed genes. So from this information, we can see that most of the genes which were mapped is something to do with the response to type 1 interferon and viral genome replication and all type 1 interferon mediated pathways. So the vaccine is triggering all the pathways that is something to do with response to defense of the viral. Because you can see type 1 interferons, the cytokines that are released early during a viral infection that activates hundreds and hundreds of antiviral genes which work in conjunction to stop the viral life cycle. So you can see as well the biological processes dot plot is just another way of representing the same information that got from the bar plot. So you can collectively see that the enrichment analysis here is demonstrating us that all the differentially expressed genes are predominantly associated with the antiviral defense mechanism. So as well, we can visualize as well what the enrichment map looks like. Okay, so the enrichment map is sort of like a graphical connection of all the genes. So basically what it's showing is it is showing the most significant genes which are interconnected, or rather which work in conjunction with each other for all the viral processes that are happening here. So we can see that when a person is introduced to a vaccine, it is going to trigger a network of genes which work in conjunction with each other to help fight the viral life cycle and create a strong response to the virus. All these processes are there to try and stop viral replication and the viral life cycle. Okay, so the next important point of core, as I have said, I'm not going to touch on cellular components and molecular function. That is too technical, and since we are just going to show you which pathways are affected, I'm now going on to KEGG. So KEGG stands for Kyoto Encyclopedia of Genes and Genomes, and it was developed by the Kyoto University in Japan. It is a comprehensive biological database that maps genes to non-molecular pathways. So just by looking at the graphics output of the KEGG pathway enrichment, we can see that all the genes are pointing to influenza A, coronavirus disease. So overall, this pathway enrichment analysis that we've done is supporting our gene ontology findings, because it is showing us that all the transcriptional changes that has happened are primarily associated with activation of antiviral immune signaling, which is something to do with influenza and coronavirus disease. Okay, so as well, the KEGG dot plot is also another visual interpretation that the genes that we may entirely associated with coronavirus disease and influenza. Okay, and then as well we can visualize the CNA plot for KEGG as well. It is also showing you that all the genes that were mapped were interconnected, but you can see the stronger nodes are at the coronavirus disease and the influenza. So it basically it is also telling us that the genes that were activated by the vaccine are pretty much associated with influenza A and coronavirus disease. So the functional enrichment analysis, it consistently demonstrated that most of the genes are primarily involved in antiviral immune response, particularly the type 1 interferon signaling. And furthermore, KEGG pathway also showed us that these genes are functioning together within a conserved antiviral signaling pathway. Okay, so I think if you are a non-technical person and you really love what I do and you have followed this video up to the end, I might also have shed some light towards what you might be thinking on how vaccines work. We have clearly seen that vaccines do not change who we are genetically. They don't rewrite our DNA. They just help the body to recognize that this is a virus, and the body already knows how to deal with foreign organisms. So vaccination just helps the immune system to activate these existing defense mechanisms more effectively rather than changing our genetic blueprint. So thank you to those who have followed to the end of the video. Please just like, share and subscribe. Thank you.